These are the results of the complementation test. The complementation test is a test for function. The cripple and the blind, which I used last week. So what you see here are a bunch of infections in a host that cannot allow an R2 mutant to grow. As you see, all these, what he called the self-test, all of these are negative. There is no phage production. All of these are R2 mutant. And the wild type is making a lot of phage. And this is a typical complementation between an R2A mutant and an R2B mutant. The first gene, the first genome on the top, is providing the normal R2B function. The bottom one is providing the normal R2A function. So in the complementation test, Benzer and Champ used two R2A deletions, and these two show that 1589 is R2A minus as expected. But in the next one, all these 5 complementation tests give a positive result, that is phage production. You may say that 220 is different from 260 or 300 in terms of, or 360 or 280. No, in terms of yield of particle per infected cell, between 100 and 300 is a normal yield. In his case, the yield were reasonably high. They were above 200. So you have a deletion that is destroying a part of the R2 locus, including a part of the B locus, and that deletion is not affecting the R2B function. Even though these beginning of the region, this beginning of the R2B gene, has sites for mutation. Okay. So then Benzer introduces the concept of sense and nonsense. Sense means a nucleotide sequence that can be translated into a protein, a regular codon. Nonsense means a sequence of bases that cannot be translated into an amino acid. That's nonsense and sense. So he realizes that, and he explains that in his mind, and we will see that next week, in his mind, this indicates that the genetic code is not completely degenerate. A degenerate code is a code where all the words have a meaning, where all the codons, all the nucleotide sequences can be expressed into an amino acid, into a protein. That's a fully degenerate code. At the end of this, Benzer realizes not only the implication for the code, but he also realizes the implication for evolution. This paper described the first protein chimera, chimeric protein. Now chimeric proteins are very common, people use them all the time. That's the first chimeric protein, a chimera between part of A and part of B. And so he thinks about protein structure in general, and he says, well, maybe if you can fuse 2 genes to make a single gene, maybe you can separate genes to make a single gene to make 2 separate genes. And one of the examples discussed by Benzer and Champ is the case of the tryptophan synthetase. The tryptophan synthetase in E. coli has a TRPB gene followed by a TRPA gene. And so he proposes that maybe during evolution there was an original gene that was a complete BA, and then BA got split. And this is one of the way evolution could work. Well, it's a nice proposal, but it turns out that the proposal is not entirely accurate, because it was found later that in eukaryotes, such as yeast or Neurospora, the order of the genes is reversed, and TRPA precedes TRPB, and the two polypeptide chains, the two proteins, are linked together by what is called a linker. This is a crystal structure of a eukaryotic tryptophan synthetase with the TRPA gene in yellow and the TRPB gene in blue. And this you can join. There's no way you can keep this structure and join B first, A next, because the C terminus of B is in this region, the N terminus of A is on the other side of the molecule. There's no way you could link them. I mean, you would require a huge linker to do that. Now, when the sequence came out, it was realized that in fact the two genes are separated by two nucleotides, a stop codon, a nonsense codon, TGA, and that this is one of the examples where one base, here, the A, serves two functions. One nucleotide is used as part of the stop nonsense codon of TRPB, and this same A residue is part of the start codon of TRPA. So the amount of information that can be stored in a single base is more than what was thought before. Now, you may think TRPA, TRPB, one enzyme, two enzymes with one gene? I mean, how is that compatible with the one gene, one enzyme model of Beal and Tatum that we've seen in the first session? That's the reaction carried out by TRPA and TRPB. You first remove a glycerol 3-phosphate, you have an indole residue, and then you add this part, which makes it tryptophan with an amino acid. Now, this reaction is carried out by TRPA and this by TRPB. In a typical enzyme reaction, you would have this IGP entering the enzyme, and indole coming out of the enzyme because it's a product, and indole going back into the active site of the second enzyme. Well, in fact, this is basically one enzyme because the indole is never free. The indole is produced here and is pushed down this tunnel into the second active site. So it's basically one reaction. Although you can write it as two reactions, but it's still one enzyme.